Hello everyone. I Kalpana Ranwal, a student of MSc Microbiology, second semester, is going to deliver a presentation on DNA structure A, B, and Z DNA. Please like, subscribe, and press the bell icon. Nucleic acid. Nucleic acid basically are biopolymers which are made up of monomeric units known as nucleotides. Two types of nucleic acid: DNA and RNA. First, DNA. DNA, which is double-stranded molecules having cytosine, guanine, adenine, thymine bases, and RNA, which is single-stranded molecules having cytosine, guanine, adenine, uracil bases. Uracil is a pyrimidine that is structurally similar to the thymine. Nucleotides. The nucleotide is made up of three groups: sugar, phosphate group, and nitrogenous base. First is sugar. Sugar is present inside the nucleic acid. 5-carbon sugar called pentose. Different type of pentose present in our body, but two types of important pentose present inside the nucleic acid are the ribose and the oxyribose. We can see the structure of ribose and deoxyribose. We can see the chemical structure. Different types of functional group attached to the carbon atom, like hydroxyl group and hydrogen atom. Deoxyribose, the carbon number 2 contain only hydrogen atom, not hydroxyl group, whereas ribose contain carbon number 2, the hydroxyl group. Second is phosphate group. Phosphorus atom in the center, to which 4 carbon atom is attached, which are negatively charged. Third is nitrogenous base. In DNA, the sugar is deoxyribose and there are 4 nitrogen base. The base are adenine, thymine, guanine, and cytosine. Adenine and guanine are purine base and two ring structure, whereas cytosine, thymine are pyrimidine base which has only one ring structure. DNA structure. DNA is a genetic material. Watson and Crick proposed a model for the structure of DNA in 1953. It consists of two helical polynucleotide chains. Two polynucleotide chains coil around the same axis to form a right-handed double helix. The purine and pyrimidine bases of each strand face inverse each other. The 10 base pair present in per helix turn, and distance between two adjacent bases is 3.4 nanometer. Helix found in 2 nanometer, and we can say the 20 angstrom. Base pair rule. Base pairing of adenine with thymine and guanine with cytosine. Hydrogen bonding. The DNA strands are held together mainly by hydrogen bond between the purine and pyrimidine bases. There are 2 hydrogen bonds between adenine and thymine, while there are 3 hydrogen bonds between guanine and cytosine. The guanine bond is therefore stronger than adenine. The guanine-cytosine bond is therefore stronger than adenine-thymine bond because of the hydrogen bond. The two strands in DNA molecules run antipolar, which means that one strand run in 5' to 3' direction, while the other is 3' to 5' direction. Types of DNA. Different forms of DNA: bDNA, aDNA, and zDNA. First is bDNA. bDNA, it is right-handed helix. Each turn contains 10 base pair per turn. The diameter of B forms is 20 angstrom. B form DNA contains two groups, major and minor. Major, which is wide and deep, and minor, which is narrow and deep. Second is aDNA. aDNA, right-handed helix. Number of base pair present each turn is 11 base pair, and aDNA forms contain two groups, major and minor. Major is narrow and deep, and the other minor group, which is wide and shallow. Third is zDNA. zDNA, left-handed helix, zigzag sugar phosphate backbone running in antipolar direction. Each turn contains 12 base pair. zDNA forms contain two groups, major and minor. Major groups is flat one and minor is narrow and deep. Thank you.